Welcome to Chapter 7, From DNA to Protein, How Cells Read the Genome. This chapter connects the material we learned about in Chapter 4 to the material we have been learning about in Chapters 5 and 6. As we discussed in Chapter 4, proteins are the workhorse of the cell. They carry out a majority of the cell's function and are approximately 50% of the dry mass of the cell. It's about time we talked about how the cells actually make these very important macromolecules. This chapter is broken down into 3 parts. In the first 2 parts, we will address how we get from DNA to RNA. Here are our lecture objectives. As always, I expect you to have these mastered before our exam. Please let me know if you are struggling with any of these objectives. There are several parts to our first topic, and we will be talking through each of these items. These are the topics that we will be covering in Part 1. As we talked about before, for a long time there was debate about whether it was DNA or proteins that pass on heritable information. Once we understood the structure of DNA, we were able to determine the process of transferring information from DNA to protein. It is composed of 2 processes, transcription and translation. While DNA is important, it does not actively make the protein. It instead serves as the blueprint, but works with enzymes to manage the process. This process of DNA to RNA to protein is the central dogma of molecular biology. This means that this process is the trunk that all other molecular biology theories and hypotheses are built off of. Let's start looking at the first part of this process, transcription. This is the opening and reading of DNA to create RNA. Each cell in the body contains 2 copies of each gene. That is not much blueprint to direct all the functions of the cell. Luckily, the cell can make multiple copies of the RNA from a single gene, allowing for the amplification of the gene, creating enough copies to create the proteins needed for cellular function. This process is monitored through gene regulation, which we will discuss in Chapter 8. Before we get too far, though, let's remind ourselves of the primary differences between RNA and DNA. The DNA molecule prefers to be a double-stranded molecule, while RNA prefers to be a single-stranded molecule. The sugars are also different. DNA contains deoxyribose, see there's a missing oxygen here, while RNA contains ribose. The last difference comes from the nitrogenous bases. DNA and RNA share 3 of the 4 bases. The 3 that are shared are adenine, cytosine, and guanine. In DNA, the final base is thymine, while in RNA it is uracil. Before we talk about the process of transcription, let's get reintroduced to how this molecule functions. There are several types of RNA produced within the cell. Each of these types of RNA perform a specific job. The 3 main types are mRNAs, rRNAs, and tRNAs. Let's take a look at each of these RNA molecules before we talk about the process of transcription. As you can see from this table, there are a variety of small RNAs that we will talk about in later chapters. mRNA is short for messenger RNA. The typical job of mRNA is to serve as the intermediate between the DNA and the protein sequence. The mRNA is truly the transcript of a specific gene. Whenever we talk about the RNA complement to the DNA molecule, it is the mRNA. After being created, this mRNA will be processed in the nucleus and sent to the cytoplasm to be translated. The next RNA that we need to talk about is rRNA. We will get to the process of translating the mRNA into an amino acid sequence in a little bit, but one of the major enzymes in this process is called the ribosome. The ribosome has a core made of RNA, specifically rRNA. In your earlier biology classes, the rRNA and the ribosome may have been used interchangeably. This is not entirely correct, but works in basic biology classes. Ribosomes are a combination of proteins and rRNA molecules. The last RNA we are going to talk about is transfer RNA, or tRNAs. The tRNAs assist the ribosome in building the amino acid sequence of the protein that is coded for by the mRNA. The role of tRNA is to code for a specific amino acid. The 3' end of the tRNA attaches to a specific amino acid. In the anticodon loop, at the bottom, so to speak, of the tRNA, there is a specific code that correlates to the mRNA, which is called the anticodon. Using these two specific regions, the tRNA, in conjunction with the ribosome, create the amino acid sequence called for in the mRNA transcript. Now that we have a handle on the structural differences between DNA and RNA, and understand the 3 main types of RNA associated with transcription and translation, let's take an overview look at the process of transcription before we get into the nitty-gritty details. The production of the mRNA transcript is the goal of the transcription process. This transcript is the complement of a region of DNA. The process looks pretty similar to the process of DNA replication that we discussed in Chapter 6. However, there are a few major differences. The first of these is that the mRNA sequence does not stay hydrogen bonded to the DNA molecules. Once the RNA polymerase has finished its job, the sequence will release from the DNA molecule, allowing the DNA to reform into the double helix. The second major difference is the length of the mRNA. In DNA replication, the entire chromosome is duplicated, while in eukaryotic transcription, only one gene is transcribed into mRNA. These genes can be copied again and again, one after the other, depending upon the demand for the product of the gene. The last difference between replication and transcription lies with the RNA polymerase. Besides the obvious, that RNA polymerase uses ribonucleotides instead of deoxynucleotides, it also does not require the primer that DNA polymerase requires. This difference can generate more errors in the transcript. However, errors in an mRNA sequence are less likely to cause damage than errors in the DNA sequence. This may explain why the two different processes occur. The goal of transcription is to generate a lot of product quickly, with errors being acceptable, while the goal of replication is to copy the chromosome with as little errors as possible. Now let's look at the machine that drives transcription. In this section, we are going to talk about where transcription starts, how it is initiated, the entire progression, and the processing of the product. Just like in DNA replication, we need to start our discussion talking about where transcription begins. After the double helix has been relaxed by enzymes to expose the regions of the DNA that will be transcribed, the RNA polymerase will randomly attach to the DNA molecule and look for a specific region on the DNA known as a promoter. The sequence tells the RNA polymerase that this is the place to begin transcription. This recognition of the promoter in prokaryotes is the responsibility of the sigma factor. This is a special subunit of the enzyme that helps the polymerase find the promoter sequences. Once bound, the sigma factor will dissociate the enzyme. After recognition of the promoter, the RNA polymerase will then open the hydrogen bonds of the bases, or the rungs of the ladder of the double helix. In this way, the RNA polymerase is performing the jobs of many different enzymes in DNA replication. In eukaryotes, the RNA polymerases need the assistance of several transcription factors instead of the simple sigma factor. The complex of the RNA polymerase and transcription factors is called the transcription initiation complex. Unlike the sigma factor, which helps guide the polymerase, in eukaryotes the transcription factors will actually sit on the promoter region which helps direct the RNA polymerase. They also help open the double helix and start to get the RNA polymerase started. Given the amount of information we have to cover in this chapter, I do not expect you to memorize the components of the transcription initiation complex. Just understand the basics of what the overall function is, where it happens, and why it happens. Moving on to the promoter region itself, we know of two specific promoter regions in the prokaryotic genome. These regions come in pairs for the polymerase to recognize. In prokaryotes, they are always 35 bases and 10 bases ahead of the region that should be transcribed. The specific location of these promoter regions help tell the RNA polymerase which direction it needs to read the DNA sequence, or, in other words, which strand of the DNA is the template strand. The sequence 10 bases upstream from the gene is referred to as the TATA box. This TATA box also exists in the eukaryote, but it is located 25 base pairs prior to the start of the gene. Well, that's it for part 1 of the chapter. We will continue the topic of DNA replication versus DNA transcription in part 2.